Hello everyone, thank you for having me here once again. My name is Alok Longodeng, a student at Ines Ruhanjeri, Faculty of Health Science and Department of Biomedical Laboratory Science. So today I'm presenting a video on applied terminology regarding to vaccines. So let's look at the table of contents. So the table of contents consists of the principles of vaccines, which is from question 16 to question 18. And preparation of vaccines, which is from question 19 to question 20. So let's look at the first question, which is what makes a vaccine effective. An effective vaccine stimulates a strong and lasting immune response, protects against infection or severe diseases, as we know, and is safe for population. So here are some key features of the effectiveness of the vaccine. So the first feature is antigen selection. So the right part of the pathogen is chosen to stimulate an immune response. Then we have immune response. A strong and lasting immune response is induced. It means a strong and immune response is needed, including the production of antibodies and memory cells. Thirdly, we have adjuvants. As we know that adjuvants are substances that are added to increase the immunogenicity. So these substances enhance the body's immune response to the vaccine. And then we have storage and handling. There should be a proper storage and handling to ensure that the vaccine remains effective until administration. Let's look at an diagram here. So this diagram, it also explains what we just talked about. First is a strong immune response, which is immunogenicity. The vaccine must be induced. A robust immune response, including antibody production and activation of T cells. So this is all about a strong immune response. Then we have proper antigen presentation. So in proper antigen presentation, the antigen should be well presented. Then adjuvants to enhance immune response, and then storage and handling conditions. It should be well stored for safety and to avoid also the side effects. Let's look at the next question. But before we should look at the next question, we should know that the examples of these factors or the effectiveness of these vaccines, we have the smallpox vaccines, which is eradicated small globally due to its effectiveness, and we have those of mumps, rubella, and plasmas vaccines. So let's look at the next question, which is question number 17, which is how do messenger RNA vaccines work? So first, messenger RNA vaccines use a synthetic form of messenger RNA to instruct cells to produce antigen and triggering the immune response. Here are some key points or mechanisms on how it works. First, we have messenger RNA introduction. So the vaccine contains synthetic messenger RNA that encodes a part of the virus, typically the spike protein. Then we have the lipid nanoparticles. The messenger RNA is encapsulated. It is encapsulated in the lipid nanoparticle, which protect the messenger RNA and help it to enter human cells. Then thirdly, we have immune response activation. The spike protein is displaced on the cell surface, promoting the antigen promotion to recognize. Let's look at the diagram here. We also have memory cell formation, where the immune system produces antibodies. It produces antibodies in memory cells. which remain in the body to provide long-term protection in the future. So here are how messenger RNA works on this diagram. It also explains what we just did, like here, messenger RNA delivery to human cells, protein synthesis, uh, memory cell formation, and immune response activation. So quickly, let's look at the next question, which is question number 18. To why are adjuvants used in vaccinations? First, adjuvants are added to vaccines to boost their immune response, making them more effective. So, in in small way, the definition of adjuvants: these are substances that enhance the immune response to the antigen in a vaccine. So here are some reasons. The first, we have immune enhanced immune response. Adjuvants help to boost body's immune response to vaccine, making it more effective. Then they promote antibody production. So the production of antibodies, which is crucial for fighting off effectiveness, as we know that, or infections, as we know that antibodies fight against infections. Then we have the encouraged T cell activation, and this brings us to number 4, which is uh, the improve vaccine efficiency. By they increase the overall of the immune response. Adjuvants make it more effective, especially in populations with weaker immune system, for example, those at the elderly age or the old ones. Let's look at the diagram quickly. To why adjuvants, like we said previously, they enhance immune response, which they increase the recognition of the antigen, they prolong antigen exposure. Uh, this they also prolong antigen exposure and improve vaccine efficiency. The efficiency of the vaccine is improved by the adjuvants, and also lastly they induce memory cell formation. So example of these adjuvants are aluminium, salts, and MF59. So it's an oil based on the adjuvant in some influenza vaccines which is MF59. So this brings us to question 19, to how do live attenuated vaccines, or how are they prepared? So at first, live attenuated vaccines are made by weakening the pathogen so it cannot cause disease, but it still triggers the immune system. It doesn't cause disease, but it still triggers the immune system. Here we have first is pathogen selection. So while we are preparing, or before, at the start point of preparing vaccines or live attenuated vaccines, so the first thing we should look at is the pathogen selection, the one we are going to use. Is it going to be a virus or a bacteria to be used? And then attenuation, which is the process of weakening the pathogen through culturing or genetic modification, where genes are responsible for virulence of attenuated are altered or deleted. And then also we look at the safety testing to ensure that the live attenuated strain does not cause any disease. We should make sure that it is not effective. And then production or grow in live attenuated pathogen in large quantities. So we have formulation, where stabilizers are added to preserve for efficiency, and then quality control. Conduct tests to meet the safety and potency standards. So here's a diagram where we are going to look at. So it also says pathogen isolation, as we already talked about. You should first select the pathogen. Is it going to be a virus or a bacteria? And then attenuation process, where we have to weaken it. Then we have culturing in non-human cells, as we say. It can be genetic or selective culture media, and then quality control and testing to make sure that it's safe. So the examples of these are MAPS rubella vaccines, which are live attenuated, and also oral polio vaccine, which is made using attenuated polio vaccines. So this brings us to question number 20, which is going to be our last question. To what are the advantages of subunit vaccines? Subunit vaccines are purified components of pathogens such as proteins or sugars that elect an immune response, providing involving the entire pathogen. So here are some. We have safety. Subunit vaccines only contain a specific pathogen, reducing the risk to cause disease. The targeted immune response, they elicit a strong immune response, especially against targeted antigens. Then we have stability, which is they are more stable than the live attenuated. There's no risk for reversion, so they are not that risky. And reduced side effects. Their side effects are reduced compared to the live attenuated vaccines. So here is a quick diagram where we should look at, where we have the stability and storage, as we already talked about. It is easier to store and transport. So we can easily store and transport these subunit vaccines. Also, we have safety, or they are safe because there are no live pathogens, so there is no risk of them causing any disease. So it means they cannot cause any disease. We also have targeted immune response. It focuses on the immune response on a specific antigen. So they have targeted. You don't just go and use any antigen. They have specific antigens they use. And then they have lower risk or side effects. So only most of the immunologist part of the pathogen is induced, reducing the advance of the reaction. So it means they have lower risk of this. And I think this brings us to the end of our presentation of this module. So thank you for listening and staying tuned. See you in the next upcoming videos.